Hello everyone, thank you for joining the session this evening. In this session, Professor Chris Yau will be delving into the world of artificial intelligence and how it can be used in cancer research to bring benefits to cancer patients. This includes his current research to find more effective treatment options for cancer patients and the seemingly endless possibilities of how artificial intelligence technology could help to advance medical research. He will also discuss how we perceive artificial intelligence in healthcare, breaking down taboos and exploring what the future may hold. Professor Chris Yau is Professor of Artificial Intelligence at the University of Manchester, a Turing AI Fellow and Director of Health Data Research UK-Turing Wellcome PhD programme in Health Data Science. His research focuses on using technology and data to give insight into how diseases such as cancer develops. He currently co-leads the Machine Learning Clinical Interpretation Partnership as part of the Genomics England 100,000 Genome Project. Before I hand over to Chris, if you have any questions, please pop them in the Q&A box for the end of the session where Chris can answer them live. And there'll also be interactive elements where you can pop your thoughts or answers into the chat box as well. So over to you, Chris.
Thanks for the introduction, Faye, and thanks everyone for joining this evening. I'm going to be talking about artificial intelligence research in cancer, and I'm delighted to be able to present this work today. So as Faye has already introduced me, Professor of Artificial Intelligence at the University of Manchester. My background is as an engineer. That's what I trained in, and then I started working in the area of mathematics and statistics when I did my PhD many years ago now. And the focus of my research is using mathematics and computation to analyze molecular information that is measured from cancers. And what I'm trying to do is to help clinicians determine what the optimal treatment regimes are for patients based on the molecular state of their cancer. So the background to this work is that it was funded by a strategic government initiative called the Turing AI Fellowship this time last year. I was one of 16 recipients at the time, and I have a particular project which is focusing on developing artificial intelligence for cancer and to help support clinicians to treat cancer. But there's a little bit of further background to this, and one of the reasons why I work in cancer research is that it's a fairly personal matter for me. The photo on the left shows me as a young boy, and this was a photo that was taken a couple of years after my father, in the middle there, was diagnosed with colorectal cancer. And in the years subsequent to that, what we discovered was that actually my family suffers from something known as Lynch syndrome. And for those who are aware of Lynch syndrome, it's a condition which causes genetic predisposition primarily to colorectal cancer, but also ovarian cancer as well in some women who have this particular condition. I was fortunate to be genetically tested in my early teens, and I don't carry the mutation that causes Lynch, but several members of my family do have Lynch syndrome. And this happened when I was young, and so really inspired me to work in this particular area. So what is this talk going to be about? First, I'm going to talk to you about the need for artificial intelligence. I'm going to motivate that need in cancer research. I'll then talk a little bit about what is actually artificial intelligence. There are many definitions. I'll talk about specifically the definition that applies to the work that I do. I'll then talk about an application of artificial intelligence in cancer, and something that I particularly work on characterizing disease progression. And then I'll end the talk by talking about some of the issues related to artificial intelligence that are widely discussed in the public. But before I move any further and kind of motivate the need for artificial intelligence, I'm going to do a little exercise with you. So hopefully nothing too taxing, but do get ready to post something in the chat in response to what I'm about to show you. So what you should be seeing now is two sets of objects, Group A on the left-hand side of your screen, and Group B, which is on the right-hand side of your screen. And what I'd like you to do, just over the next sort of 10 seconds or so, is to look at those pictures attached to Group A and attached to Group B, and to memorize which type of object belongs in each group. Great, that's 10 seconds. So I'm going to change my slide, and I'm going to show you a sequence of 5 new objects. And what I'd like you to do is to classify those objects into either Group A or Group B. So here comes the first one. Which group does this object belong in? Is it Group A or Group B? If you want to, please post it in the chat. Okay, I'm going to change slides and show you a second object. So which group does this object belong in? Group A or Group B? And the third one, Group A or Group B? And the fourth object, Group A or Group B? And the final one, Group A or Group B? Thank you. So hopefully Faye has been watching the chat and has scribbled down what the majority vote has been for each of those 5 sequences.
Yeah.
Faye?
So for the first one, it was a mix of B and A. It's kind of nearly a 50 split. I think a couple more Bs. Mm. Number 2, everyone said B. Number 3, everyone said B. Number 4, everyone said A. And number 5, most said B and a couple said A. Okay. So my prediction would have been that most people would have said this, A, B, B, A, B. And I think that broadly matches what you've specified. 2 to 5 it does, but 1 it was a bit more split. Yep. A to B. Probably because of the first one. It might have been a bit of a people need to get adjusted to the exercise. So I'm now going to repeat the exercise again, and this time I'm going to show you 2 groups of objects again, and then we'll spend 10 seconds just memorizing what type of objects belong in Group A and Group B, and then I'll show you another 5 shapes. So the left-hand side is Group A, the right-hand side is Group B. If you can remember what type of object belong to Group A and Group B, that would be most useful. And then I'm going to show you another sequence of 5 objects. I'll go a little bit more quickly this time, now that hopefully everyone's used to what I'm doing. But I shall now show the first object. Group A or Group B? Second object coming up. Group A or Group B? Third object. I think I've changed. Third object. And now, actually, this is the 5th one, sorry. I must have repeated one. It's the 5th object.
Great. And Faye, what was the consensus this time to be?
So for number 1, the majority was B. Number 2, everyone said B.
Yep.
Number 3, most people said B. Number 4, the majority said A. And number 5, it was A.
Okay. 4 and 5 may have been mixed up because I went through 3 and 4 quickly. So, and that is as expected, because what I'm going to show in the next slide is that the first 4 objects were B's. In fact, the second and third one were actually identical. That's why I didn't actually realize I'd changed slide. And the 5th one was an A. And the purpose of this exercise was to show you that actually I had essentially shown you 2 groups of objects which were exactly the same on both instances. The only difference was one was in color and the other one was in monochrome. But when I showed you the pictures, the objects in color, the natural inclination for you, the audience, and would be for myself as well, would be to classify these objects using color. And that's what you did. And that's why you ended up with this roughly classification A, B, B, A, B. But when I turned off that color, your visual system kicked in and recognized that the 2 groups were now separated by shape. And you switch your classification to B, 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 and A. And what this example is designed to show is that even this very simple situation where I just had 2 groups of objects where there was just shape and color informing both the 2 groups, it was relatively easy to come up with a situation where, as a human being, it was a bit ambiguous as to what you might classify by. If I showed you the color, you gave one classification. If I took away the color, you would classify by shape and end up with a different grouping altogether. So there are layers of information in this particular example, and depending on which layer of information you are using as your dominant form, you would have come up with a different type of classification. And if we take that and extend that to more complicated types of data that I'll discuss later, this then presents us with a challenge. How do we solve problems like this in a very systematic way? And the need for AI here comes from the fact that what we might like to do is come up with a computer program that solves problems like this and that potentially go beyond human capability. So when I showed you the color, you only recognized the color and you kind of ignored the shape. What we might like to do, in fact, is actually have a computer program that doesn't necessarily only use color, but automatically is able to take into account and recognize that shape is also important for this kind of classification task. And so the need for AI comes when we're tackling problems which may be too hard or too large for human interpretation, where there might be important factors such as making decisions faster than human beings can normally make, and to potentially produce decisions which are more consistent and reliable. So it doesn't matter whether we're looking at this problem at 3 a.m. in the morning or first thing in the, after breakfast, and we're not affected by tiredness and things like that. But also finally, to also, as this problem illustrates, to consider different dimensions and depths of information in the information that we're presented that as human beings, we may not be able to decipher readily. So what does this mean for cancer research? Well, there's a kind of direct analog and analogy with many kind of typical cancer research experiments. So this is kind of an illustration of a typical experimental study that we might do in cancer. We may be interested in examining a particular hypothesis or question. Suppose we're interested in evaluating the effectiveness of cancer treatment, whether it's a new cancer treatment or an existing one. And the way we might do this is to gather groups of patients, some of them who completely respond or react very well to that particular cancer treatment. And then we might gather a group of people whose response is not so good, or potentially there is no response to that cancer treatment. And the typical question might be to look at those two groups of people and to ask ourselves, what is different about these groups of people? But before we do that, we might make a series of measurements. And in the work that I do predominantly, and hopefully in the future, this will become increasingly prevalent, is that we make molecular measurements. So we're going to use technologies that study all the sort of chemical molecules that might exist inside your cancer and which might indicate differences between people who respond to certain treatments and people who don't respond to certain treatments. So we might be interested in measuring levels of different proteins inside your cancer. And here's just an illustration where I could have measured 16 proteins, and the different colors correspond to different levels of those 16 different proteins. And I could do that for people who have a complete response to treatment and for people who don't have complete response to the treatment. And what I'm interested in here is comparing the protein measurements across these two groups and to determine what differences exist exist in this protein and these protein patterns. So the fundamental kind of mathematical problem or computational problem is not too dissimilar to that exercise I showed in the beginning of this talk. What we're interested in is comparing two groups. Each group is represented by a set of measurements. In this case, each patient has 16 measurements attached to them. And I'm interested in spotting the difference between protein levels between people who have no or partial response on the left and a complete response on the right. So which proteins or set of proteins look different between these two groups. Now, for those who are extremely sharp-eyed, you'll probably notice that there are two proteins in this sort of made-up example of mine, which are different. But it's quite difficult to decipher. Probably most of you wouldn't have been able to observe that, not without looking quite extensively and for a very long time. And certainly, if we measure more and more things, so there's certainly more than 16 proteins inside human cells, if we measure potentially thousands or millions of different types of molecular properties within each patient, we wouldn't be able to do this spot the difference exercise. It's just too much information for our brains to kind of interpret and to identify. So we need an algorithmic solution to solve this particular problem. And this is becoming increasingly important because molecular profiling of cancer using modern technologies like DNA sequencing are allowing us to measure a very vast number of molecular properties in cancer. So it's quite normal for us to consent for patients to take part in studies where we now collect tissues. Those tissues can go for direct DNA sequencing, or we can even split those tissues up into individual cells themselves and then to analyze each cell on its own. Now, the result of all this, once we sequenced all this material, whether it's bulk tissue in bulk or in single cells, is data, information, and lots of it. So if there are, for example, 30,000 genes that we can measure, and we do this across 10,000 cells, then that is an awful lot of information, millions and millions of data points. And there's no prospect of us being able to manually analyze that data. It has to be done algorithmically and with computers. So the need for AI in cancer is becoming essential. It's an essential tool for helping us to decipher this type of molecular profiling data, which gives us so much more information than other types of technology that have been used to study cancer in the past. And what artificial intelligence is helping me to do is to kind of systematically interrogate vast amounts of information that we collect on the cancers, and also to potentially identify biology that we may not even be aware of yet. And the second point is particularly important, is because when we compare these groups, most of the time, we actually don't know what might be different between these two groups of patients or mice or cells that we might be interested in. So unlike standard computer programs, artificial intelligence systems are designed to be a bit more flexible and adaptable. We can't pre-program these systems to know what they're going to find that we might need to teach them rules that they can use to find patterns which differentiate between two or more groups of people. So I'm going to illustrate this with an example based on work that myself and my research group have been doing, which is in the area of modeling cancer progression. So most of us are aware, either because we've been patients ourselves or family members or friends who have suffered from cancer, is that it's typical to talk about cancer progression in terms of staging, and there are different schemes or systems for staging or telling us how advanced the cancer is. And the clinical staging scales typically tell us if it's early stage that the cancer is relatively localized and highly treatable, or if it's a late stage cancer, that cancer is invaded neighboring tissues and may have even metastasized or spread across different parts of the body. Now, conventionally in clinical practice at the moment, the staging or characterization of what stage a cancer is at is done using a multitude of different types of tests and criteria. So a pathologist might look at images under the microscope of our cancer tissue and grade it. They might assign a score based on the size and number or shape of our cancer cells. There might be medical imaging, so X-rays or MRIs that tell clinicians how advanced the cancer is and whether it has spread, as well as routine tests, for example, blood tests, counting the number of cancer cells or white blood cells that all enter in to the criteria for staging different types of cancer. But what we're interested in is adding to that arsenal of current existing staging technologies is molecular profiling, or for example, DNA sequencing. So another way by which we can kind of measure how advanced a cancer is, for example, by taking the DNA from a cancer or cancer cell, sequencing it, reading that DNA sequence, and then identifying how many mutations exist in that cancer. And the assumption is that the more advanced the cancer is, the more mutations it might have. So by counting the number of mutations or other kind of molecular changes, we might be able to give a more accurate answer of exactly what state your cancer might be. And in future, it might be the case that actually we do away with some of those classic measures of disease progression and entirely only use the molecular profile of our cancers to measure the stage of disease. It gives us just so much more resolution and information that the classical measures may become somewhat redundant. Now, assuming that is the case, and knowing that we know that cancer is an evolving and progressive disease, that over time it changes, how might we want to use this information to help us better design treatments? Now, without intervention and kind of treatments, a cancer will undergo a series of molecular changes. Its molecular behavior at its early stages is very different to its molecular properties at a late stage. And this picture on the right here shows just a cartoon illustration of what might happen inside a cancer. So as it progresses, so molecule A might increase its level of activity as the cancer progresses. Molecule B might go in the other direction, actually turns off as the cancer progresses. And molecule C illustrates a type of activity where that molecule turns on for a period during the cancer's life and then turns off again. So it exhibits this sort of temporal or transient behavior. And the assumption is that here that the patient's outcome will therefore be dependent when they're treated, will be dependent on an interaction between the exact molecular makeup of the cancer at the time of the treatment and then the type of treatment given. And the goal of what people call precision medicine is to try and ensure that we make sure that the molecular makeup of the cancer matches the type of treatments that we give. So that's a really kind of fantastic goal to have, that we can design treatments based on exactly what the molecular properties of your cancer are, and therefore be able to target the exact molecular mechanisms that might be at work inside your particular cancer, and therefore hopefully promote better outcomes or even to cure the disease. But in terms of the research that leads to that final solution, we have a fundamental problem here. And the fundamental problem is that we can't access the past or the future of any particular cancer. So people who are exhibit symptoms of cancer will only obviously encounter the health system, see their GP, for example, once those symptoms become severe enough that they need to see a clinician. They'll then undergo a series of tests and become diagnosed. And at that point, we may gather a tissue sample, for example, to do our molecular analysis or other kind of clinical tests. But what that means is that we won't know anything about how that cancer evolves. We won't know anything about the history of that cancer, because the patient obviously wasn't feeling unwell in the past, and therefore didn't need there wasn't a need for any investigation. So we can't see what happened to the cancer in the past, so we don't know what sequence of events, molecular events, might have led up to the creation of that cancer. We also can't see the undisturbed future of that cancer either. Of course, as soon as the patient is diagnosed, they will need to be put on a treatment plan and treated for their particular disease. But once treatment starts, the kind of natural history of that cancer, natural progression of that cancer will be disturbed. So we won't see what the cancer might have looked like had it been left untreated. And why is this kind of why does this pose a challenge in terms of research? What it means is that we can never see enough information about the history or the future of undisturbed cancer to help us understand what advanced disease looks like, and therefore how we might best treat advanced disease. So the challenge we face is that when a patient is diagnosed, we only see what their cancer looks like there and then. We can't see its past, and we can't see its future had it not been treated. But it'd be useful to know that information in order for us to understand what the natural history of a cancer is, so then we can treat that cancer at different stages of its life in an optimal way. So what might we do to overcome this fundamental problem? So, as I said, a patient presents themselves at diagnosis. We can't see the past history of that cancer, and we won't see the future untreated state of the cancer, because that patient has to be put onto a treatment pathway. But what I can do is take information from another patient who's been diagnosed with a similar or exactly the same kind of disease, but perhaps that patient is at an earlier stage of disease. Perhaps they came in to see their GP earlier and were diagnosed at an earlier stage. Now, if those two people have the same kind of disease, but one comes in a bit earlier and is diagnosed a bit earlier, that green person gives me some information, maybe not exact, but some information about the potential past history of my patient here in red, because they came in at an early stage and they are having the same type of disease. And similarly, if another patient, in this case, the blue patient, comes in and is diagnosed at a later stage of disease, their molecular profile may tell me about the potential future state of my cancer if it was left untreated. Now, the idea here is that if I collect enough people, then I can use information from across a series of patients to work out a natural cancer progression timeline. So if I collect a cohort of patients, I might be able to work out the timeline of different cancer states as the cancer evolves from the information across this cohort of patients. And this is using the fact that every patient has a unique molecular unique molecular state at diagnosis. Not everyone doesn't exactly present for diagnosis exactly the same time. So if I collect a group of patients, it's very likely they will span a spectrum of disease states as measured using these molecular profiling technologies. But there's a slight problem here is that so far I've assumed that I know what the ordering of my patients are. I know how to order them from early to late stage. But this is a bit of a chicken and egg problem. The fact is I don't know what the order of my patients is yet. I'm trying to work that out from their molecular profile. So there's a sort of mathematical or computational problem here to solve, which is what is the right order of patients to use from my patient cohort to allow me to figure out what that cancer progression timeline might be. And the idea of using artificial intelligence here is to be able to search through all the different possibilities of different orderings of these patients to see which particular ordering makes sense. So I could try one ordering here where I put the yellow person first, the green person next, the red person following that, and the blue person and the slight gray person. But if I use that ordering and then I, for example, look at molecule A from their molecular profiles, what I might find with that ordering is that molecule A goes up and then down and then up and then down again. And I might think that that type of behavior for molecule A is a little bit unusual. You know, why would it be going up and down in such an erratic fashion? So I might say that that particular ordering seems unlikely because molecule A behaves in such an erratic way. I might prefer the second ordering here shown on the bottom right, where now I have yellow, red, green, gray, and blue. And now molecule A looks like it's just increasing as I move between each of these patients. And that seems a bit more believable as a type of behavior that this molecule describes how this molecule changes as the cancer progresses. It's increasing in activity as the cancer develops. And so AI is going to be really useful because if I have hundreds or thousands of patients and potentially tens of thousands of molecules to sift through, this ordering problem just can't be done manually. It has to be done using a computer algorithm. And this is what we're using to solve this particular problem. So what we've done in recent years is to develop artificial intelligence algorithms that solve this type of sorting problem. We take molecular profiles from groups of patients. We use this artificial intelligence to sort those patients into an order. And from that order, we can then extract a molecular trajectory or timeline that characterizes the sequence of molecular events as the disease goes from an early to late stage. And an example here is shown in the bottom right here with breast cancer, where we have two groups of patients, some with estrogen receptor positive status of their breast cancer and some with estrogen receptor negative. And what we show is that actually although classically, these patients might have been classified as having the exact same stage of disease. At the molecular level, actually, there is a spectrum of disease, and these patients are following two different timelines or trajectories, but ultimately, both trajectories converge towards a common molecular type. And this is revealing much more higher resolution information about how breast cancer evolves. We're also doing similar things in ovarian cancer, and I work very closely with a close colleague of mine, Professor Ahmed in Oxford, who runs an ovarian cancer research lab in there, which is also funded by Ovarian Cancer Action. And what we've been doing there is very similar to what we've been doing in breast cancer, taking in this case, multiple tissue samples from the same patient before and after chemotherapy, profiling these tissue samples with molecular technologies, and then trying to work out why chemotherapy is quite effective in some patients and less effective in other patients, where effectiveness is determined by what's happening at the molecular level. And what we're trying to do here is to work out these cancer progression sort of maps and timelines to identify when standard chemotherapy induces a positive response in patients and not in others, and whether it depends, whether the positive response depends on what stage you administer that chemotherapy in these patients. And ultimately, what we hope to do is that by designing these artificial intelligence systems, we might come up with an entirely new way of characterizing cancer and treating it. And some people who work in my particular area call this something known as a learning cancer health system. What we might want to do is for patients to come in, they undergo molecular analysis or profiling of their cancer. We're going to use artificial intelligence to process that data to answer the question of what direction is the disease going in, what stage is the current disease that's currently at, and what direction is it going in. And by knowing that information, the clinicians can then determine whether, from a portfolio of different treatments, which treatment is going to be most appropriate. You know, if a person is following the red direction, then actually if experience shows that treatment A is perhaps the best solution, or perhaps if the disease of the patient is following the blue direction, then perhaps treatment B is the optimal treatment here. And potentially even further, going even more futuristic, depending on the state of the cancer, we might be able to even engineer or design customized treatments. And this, for people who are aware of immunotherapy, is one of the research directions in immunotherapy, using the state of the patient's disease to design and engineer immune cells that will then attack their specific cancer cells. But furthermore, rather than just using it as a one-way thing just for individual patients, we might feed the outcomes of this data and the predictions and the treatments and the outcomes from this cycle and this particular patients, feed it into our database of patients, and continually learn from this database, which is continually being added to, and learn from the past. So if treatment is ineffective for a particular patient, we can add that to our database, and our artificial intelligence will adjust to account for that. And when a new patient arrives, perhaps we will not use that treatment again under those particular circumstances. So the idea of this learning cancer health system is that we can start to maintain this kind of cycle where we learn as each patient enters into the system. And this relies on the availability of high resolution molecular analysis technologies, as well as coupling that with the artificial intelligence software systems that make best use of that information, past and present and future as well. So that's about the kind of cancer research, but I wanted to end this talk by talking about some of the common issues which are raised by the use of artificial intelligence systems, or the development of artificial intelligence systems, particularly in healthcare. And the common questions are, I've summarized here, these are 5 of the most common questions which many people ask of this particular topic. First of which is, will AI replace humans? And this is generally a question which is related to our feelings about the automation of decision-making and potentially taking human interpretation and responsibility away when making decisions. The second common question is, can we explain what AI does? AI is being applied to these very, very complex problems and it's doing all these things with very complex sets of information. Can we explain actually what it does? Why is it that it is predicted that the cancer will be doing this and recommended this type of treatment? And this type of debate is generally around the issue of transparency and potentially liability. If an AI is making a decision and it can't be explained, or who takes the responsibility for that potential decision. There's also many questions about whether AI is safe. That's an obvious what that means. It's about risk and safety of using these systems. There's increasingly a lot of discussion about whether AI is biased because it uses information that's gathered on patients. And if I have overrepresentation of certain types of people in my database, does that cause the artificial intelligence system to be biased towards certain groups, for example. And this is generally talking about the issue of fairness and potentially discrimination, whether AI potentially solves that problem or exacerbates that particular issue. And finally, there's also an issue related to the integration and adherence to human and social values. Is AI trustworthy? It's hard to talk about an algorithm being trustworthy, but the way we design it as human designers might raise that as an issue. I won't, as part of this talk, be able to talk specifically about each of these particular areas, as there's a huge amount of material to go through and many kind of issues there. But what I will summarize is with the following. And what I'll say is that it's a bit like parenting. Artificial intelligence algorithms are ultimately designed to implement a set of learning rules. And these algorithms apply these rules to the real world information it receives and adapts its outputs to meet objectives that we as the human designers set. And it's a bit like, the reason why I say it's a bit like parenting is that with AI algorithms, the choice of ground rules that we set can lead to more or less desirable characteristics. These algorithms, unlike sort of classical software, are more flexible and adaptable, as I said at the beginning of the talk, and their behavior is not completely pre-specified at the time of design. There's flexibility for these systems to adapt. But what we do is set the ground rules for how these algorithms learn. And if there are mistakes or unintended consequences of our kind of ground rules, that may lead to unexpected behaviors. And so the rule I kind of set myself for developing AI systems is to ensure that, if they are deployed or if we're testing them, is that there should be continued monitoring after deployment to make sure that the behavior of the system is as we expect and is constantly being evaluated. The second point I wanted to kind of make is that the depth of information that we collect and use within these AI systems is really important. There are many things that we can measure that are associated with cancer outcomes. I've only concentrated on molecular stuff, genetics, collected from cells and tissues, but lifestyle and diet, your levels of fitness and activity and general health, as well as socioeconomic status, ethnicity, and gender, and where you live, are also factors which are associated with different types of cancer outcomes. And what I've kind of shown you is that it's very useful to be able to design artificial intelligence tools to allow us to probe these different layers of information and to analyze very large quantities of this kind of information. But in the real world, some of the measurements of some of these factors, some of them genuinely can cause cancer, but some of these factors don't cause cancer at all, but they seem to be linked to cancer because they become entangled with factors which actually are causing cancer. And disentangling which factors genuinely cause the disease and which are factors which don't cause disease, just happen for whatever reason to become linked to causes of disease, is an important factor in the design of these algorithms. So an awareness that different factors might become entangled is really critical in the interpretation and design of artificial intelligence systems. And finally, what I'm really trying to say is that artificial intelligence really is just like, in a healthcare setting, is just like any medical advice that we might build or engineer. It needs to be made in following good practice. And 4 rules of thumb that I recommend and suggest, and I always encourage patients in the public to kind of hold designers to, is that AI should be a solution to a problem, not a solution in need of a problem. And what I mean this is that it's kind of potentially financially lucrative or cool to be working in artificial intelligence at the moment, but for healthcare or medical device, the artificial intelligence should really be a solution to a problem, not just a thing that we want to have because it seems exciting. It's absolutely crucial that that technology is backed up by reliable evidence of patient benefit. It's possible to design all sorts of technology to tackle different types of problems, but there needs to be evidence that it provides patient benefit, and a significant amount of patient benefit to potentially justify the costs of implementing that particular system or solution. It needs to integrate within the existing healthcare pathways. It can't cause our stressed health system more work, or if it requires the design of new healthcare pathways, the designers need to tell us how to build that new healthcare pathway and to consider all the factors that go into changing the way we might treat and care for cancer patients. And as I said before, these systems must be subject to ongoing performance monitoring to make sure they work the way we intended and there aren't any unintended performance issues. So I'm going to end my talk now by concluding that artificial intelligence, I believe, is really an indispensable tool in modern cancer research. It's absolutely critical for large data processing tasks which are associated with new measurement technologies, particularly those which measure the abundance of different types of molecules on a cellular level, in fact, possibly. It's important as an important tool for ensuring that we interpret information consistently and reliably, simply because it's not possible to manually process this type of information as a human being. And if we design these systems well, it could allow us, in conjunction with developments in these molecular technologies, allow us to really fundamentally transform future cancer services. And I just wanted to end by, you as the audience, as public or patients, can really help this research endeavor by maintaining a critical and vigilant stance about the benefits of the technology, promoting the exciting parts, but also making sure that we, as designers, demonstrate the real benefits and highlight the evidence that supports the benefits of this technology. So I just wanted to end by thanking you for joining, and I'll be happy to take any questions.
Thank you, Chris. That was a fascinating overview of your work, and I like how you explained how you'll get over the conundrum of not being able to see the past and untreated future of the tumour and how AI will help with that. So I really liked how you explained that, and also I'm sure the viewers really appreciated how you broke down those common questions around AI, but you could spend hours talking about those. The more you talk about it, the more questions probably would come. So thank you very much, Chris. We have some really good questions through, so I'll start to feed those through. So someone asked, is the progression of the concentration / behaviour of the molecules affected only by stage, or are there other patient characteristics, for example age, that may affect it as well?
Yes, so there are a multitude of factors. Age is one of them. It could just be based on your gender or background ethnicity as well as all those factors that I highlighted towards the end of my talk, your general levels of activity. So I talked mostly about a relatively stylized situation where you just have molecular status and disease, no disease, response, no response. But actually, in fact, as part of our research, we're trying to factor in all these other background factors as well.
Great, thank you. Someone else has asked, what algorithms do you use in AI?
It's hard to kind of distill that very briefly, but a lot of the work that I do is broadly characterized as what we call probabilistic algorithms. And what I mean by that is that we start off with the premise that everything that we're trying to learn about, there's always some uncertainty attached to it. We can never know what's truly happening in the world. We take measurements which have a certain tolerance, and we can make inferences based on those measurements about the true state of a person's cancer or their future prospect, prognosis, etc. And the approach we take on this kind of probabilistic approach takes that into account. So everything that we estimate always comes with it some measure of uncertainty that can then be used to determine whether the output of that algorithm is really sure about what statement it's making, or actually it's making a statement, but it's not quite sure, if that's going to be true or not. And that can be quite useful for interpretation.
Great, thank you, Chris. Someone's asked, how can you take into account the information provided by patients who die after diagnosis, but before receiving a treatment?
So for people who die before receiving treatment, we can still use that information. We would essentially, what we won't get from that patient is obviously the information about what happens after treatment, but we can still enter it into our kind of database of patients. We just label that person as only having a diagnostic sample. So their contribution to the overall information content to the database is perhaps less than someone who has multiple measurements before and after treatment, have recurrence, etc. But they're still making a contribution because their molecular profile is a real measured molecular profile. If it's consistent with other patients, fine. It's just telling us what other patients were telling us. But occasionally, there are subgroups of patients who behave a little bit differently to everyone else for whatever reason, and looking for these potentially rare subgroups of patients is incredibly valuable as well. Again, to tell us what might be different. So when we design these systems, they're designed assuming that patients follow a particular pattern. It's always useful to know if that's patients who might follow a slightly different pattern exist and what that might look like.
Great, thank you. And we've had a really good question. Where databases of patients' information around tissue samples and blood samples is being gathered by other research projects, is it possible to share this rather than to duplicate it and cooperate between the potential silos?
Yes, I mean, in the ideal world, there will be a mechanism by which all researchers can undertake appropriate processes to share and reuse data. I think both in the UK and internationally, we're still aspiring to that goal. There are multiple obstacles. There's the kind of legal side and consent side to that. But also, as part of the silo, reason data is siloed is often because we don't have the infrastructure to share data. So, data might be sitting on a hard drive on a computer in someone's laboratory, which is not connected to the internet. And there's no way for someone like myself to be able to access that data without physically going there and transporting it out myself. So there are initiatives happening both in the UK and internationally to build infrastructure that simplifies data sharing, as well as initiatives in terms of culture as well, to improve research culture and scientific culture to encourage more data sharing. Historically, it hasn't always been the case that it's advantageous for scientists to share data, but that's something that most research funders are now clamping down on, saying you can't hold on to this valuable information.
And that's something that charities can help out with as well, is trying to get people to join up to share their data. But yeah, it's difficult, especially clinical data in the NHS. It's stored on different systems, and I think people are working now to try and join up those silos and to make more fantastic research happen like this. So that's all the questions that we had. Thank you so much, Chris. That was fascinating. And yeah, I'm sure, where can people go to find out more about your work?
If they go to follow if they use Twitter, they can go there and find my personal homepage from there. Else, if you just Google my name, I'm usually towards the top of the search lists.
Great. Well, thank you very much, and thank you to everyone for joining us this evening. And I hope you have a good rest of your week and join us soon for another research webinar. Thank you.